Welcome, everyone, to the Winter Quarter CDM Research Colloquium series. For the seminar this week, it's my great pleasure to introduce Dr. Robert Tell, who is currently the Vice President of Bioinformatics at Tempus Labs Incorporated, located here in Chicago. Dr. Tell holds a bachelor's degree in cell and molecular biology from University of Illinois, Urbana-Champaign, PhD in pharmacology from University of Illinois, Chicago, and also was a postdoctoral fellow at Northwestern University. Before joining Tempus Labs in 2016, Dr. Tell was a senior bioinformatics scientist at Abbott Laboratories, which is also headquartered in Chicago. A little bit about Tempus Labs. It's a technology company that has built the world's largest library of clinical and molecular data and an operating system to make that information accessible and useful for patients, physicians, and researchers. The mission of Tempus Labs is to usher in an era of personalized medicine. Dr. Tell is at the helm of bioinformatics operations in Tempus Labs, and I'm really glad to have him at our colloquium to share some of the cutting-edge research work happening at Tempus Labs. The title of his talk today is Liquid Biopsy Testing at Tempus and Modalities for Therapeutic Intervention. Welcome again. Thank you so much for being here, and please take it away.
Well, thank you so much for that great introduction. It's a pleasure to be here. Basically, today I was hoping to talk about liquid biopsy-based oncology testing, some of the technical challenges, and the methodologies we employ at Tempus to attempt to overcome these technical challenges. So first of all, I want to go into a bit of detail about what we're going to talk about. First, what is liquid biopsy? Why do we use it? Why is it important? What are the current challenges in liquid biopsy testing? How does the Tempus assay that we've developed address these challenges? And then we will review the data from a 1,000 patient trial cohort where we employed this assay to perform liquid biopsy genomic profiling. So a lot of the work I will be talking about will be from this paper here, Validation of a Liquid Biopsy Assay with Molecular and Clinical Profiling of Circulating Tumor DNA. Please review the author list. They're the ones who did the real work. I am just the manager. So yeah, I'm very happy to share their excellent work. So first, what is liquid biopsy? As we all know, when you get a blood tube, you get 3 phases once you spin it down or let it sit on the countertop for a while. You get a plasma phase, a buffy coat, which is your white blood cells, and erythrocytes, your red blood cells. So what we realized about half a decade ago now is that there is cell-free DNA present in the buffy coat sample, the plasma itself. And when you have cancer, there is cell-free DNA derived from the patient's tumor that is present within this buffy coat sample. So what you can do is a variety of sequencing-based approaches to interrogate the cell-free DNA for cancer-based biomarkers or cancer-derived biomarkers. So here's where you see a variety of methods employed for ctDNA-based analyses. You see things like monitoring, tumor burden analysis. ctDNA correlates linearly with tumor size, so you're able to do some interesting tracking there. And the different methodologies that can be employed: ddPCR, NGS, next generation sequencing, or whole genome sequencing or whole exome sequencing. So in the context of liquid biopsy, there are a variety of different, as we showed earlier, modalities of intervention. The first one, of course, you've probably heard a lot of news around the company Grail that recently released their Gallery test, which is an early cancer detection test that uses methylation profiles to detect a variety of different solid tumors from patient plasma. There's also surveillance for micrometastatic disease, point 2, that we call that minimal residual disease or molecular residual disease analysis. That is to say, we know the patient has cancer. We're trying to get a proxy for recurrence or response, usually in the curative intent treatment setting. That is, patients where we're trying to cure the disease rather than manage it, so earlier stage disease. And then in the last phase, there is treatment selection and response monitoring in patients with metastatic disease. That is to say, In stage 4 patients, we want to use biomarkers from plasma to try to inform us on what therapies to give the patient, whether they are responding to existing therapies, whether they're developing resistance mutations that could reduce the efficacy of current therapies, or whether we should switch therapies. So today we're going to talk about number 3, the Tempus assay, the XF assay we call it, is a therapy selection assay for primarily for patients in the metastatic setting, that is to say stage 3 or 4 disease. So here we see some very excellent work from 2014 where they're showing ctDNA, tumor-derived ctDNA concentration directly correlating with tumor size on radiography. So they take a blood sample, extract the plasma, and analyze the plasma for biomarkers, and then they do a CT or scan or MRI, and they map the size of the tumor to the amount of ctDNA detected. Usually this would be described in mutant molecules per milliliter. So you have a genomic alteration that you know is associated with the cancer, and you measure the, for lack of a better term, percent of molecules you pick up out of the total number of molecules that could that have that mutation. And if that percentage goes up or down, you correlate that directly with tumor size. So what this means is, of course, we can use this for serial monitoring to evaluate tumor dynamics. We can, of course, use this for predictions of response to immuno-oncology drugs, which, of course, are very becoming very much a mainstay of treatment in the metastatic setting, and detection of emerging resistance mutations. That is to say, mutations that block the effectiveness of targeted therapies. The most common example, if anyone's familiar at all with non-small cell lung cancer, there's a specific drug, tyrosine kinase inhibitors, that are targeted against the epidermal growth factor receptor gene, EGFR. Gain-of-function mutations in that gene can drive oncogenic signaling pathways. We treat it with anti-tyrosine kinase drugs. This is very effective until the patients inevitably develop a resistance mutation to these tyrosine kinase inhibitors. Detecting that resistance mutation is critical because then the therapy is no longer effective. So this is one example of how liquid biopsy is quite useful for these patients. So we don't have to essentially re-biopsy the tumor. So again, to that point, you can see these graphs on the right where we're showing phasing therapies with tumor response dynamics. You can see a number of different patients published in the this is a TRACER-X study where they're showing treatment in the colored bars, and ctDNA concentration are the lines and dots. So we're seeing both patients responding effectively to therapy, patients not responding to therapy, and patients relapsing as we observe higher levels of ctDNA present in circulation. So I apologize. I keep looking at this to the side. My slides are on a different monitor. But yeah, we can clearly see the value of this monitoring as an orthogonal method of assessing patient response as we change therapies, as we evaluate course of therapy, as we try to manage these patients in the course of their disease. So to that end, go a little bit over the advantages and some of the challenges of liquid biopsy that we still are facing. We've clearly seen that there's a lot we can do with this technology, but there's still a lot of technical difficulty. The biggest advantage is there's no need for a tissue biopsy. You don't have to do surgery on a patient. You don't have to stick a giant core needle into the tumor to extract tissue. You can just get a blood sample. You also get to profile all lesions, all cancer lesions simultaneously. That is to say, they're all those cells are dying. All of them are releasing DNA into the circulating blood supply. Whereas when you do a biopsy, you're only getting the site you biopsy. And if you're in the context of metastatic disease, you're only biopsying one of many sites, and clonal dynamics occurring in different sites may be driving a disease state. And that's something that we find to be an advantage of liquid biopsies. You can detect all these different phenomena. It's also a good proxy for therapeutic response. That is to say, how much ctDNA there is tells you a lot about how the patient is responding to a therapy. The remaining challenges are negatives are not informative. if you don't detect something, that doesn't mean it's not there. It just means that the patient may not be shedding ctDNA. It may not be detectable. And a significant percentage of patients do not shed ctDNA into the bloodstream for reasons we don't quite understand. There's also the biggest one we'll talk about here is there's a high degree of technical difficulty, both on the chemistry side and on the computational side. Of course, I imagine you're all more interested in the computational side, so we'll talk more about that. But suffice to say, significant challenges remain in both areas, and in developing a bioinformatics assay. Both of these challenges are, or a next-generation sequencing assay that is both a combination of bioinformatics and wet lab chemistry. Both challenges are significant and can be insurmountable in some cases. So here is one of the clear technical challenges here. For those who are unfamiliar with next generation sequencing, essentially what we do here, this is a good visualization of a DNA sequencing pileup, we call it. So basically we see this saying simulated mutations. What we're doing is serially resampling the same exact genomic position over and over again and doing DNA sequencing of that information. So we will sequence the same region of the genome thousands or tens of thousands of times to try to resolve these mutations present in the bloodstream. That we have to do this because these mutations tend to be a very small percentage of all circulating DNA. This is because essentially most of circulating cell-free DNA is immune cells, but all cells within the body are undergoing apoptosis. They're dying. They're releasing their DNA into the bloodstream. So while the cancer cells are doing this as well, they may be doing it at a relatively small rate compared to the entire rest of the body. So we have to resample the same area thousands to tens of thousands of times to be able to detect these alterations. And here's what we're seeing, basically requirements around depth and a number of other target parameters around the methodology of sampling. In this context, I'll just go into it a little bit. It's called a hybridization capture technology. If you all remember your biology, DNA is complementary, has reverse complement. So it's possible to enrich a DNA fragment by making the reverse complement of its sequence. So you can engineer a DNA fragment that will bind to the DNA fragment you're interested in sequencing. And you can essentially enrich those by pulling them down with engineered DNA fragments. So it's a really cool process. And it results in these sort of enriched genomic regions that we think are informative from a biomarker perspective. So here's the problem. High-depth sequencing generates a very high error rate. The standard error rate for sequencing is 1 times 10 to the negative third. And if you're sequencing to 10,000x, you can immediately see how detecting low prevalence or low percentage events becomes very challenging because of the signal to noise boundary. So this is an example of how you get errors introduced either through the sequencing process or through random events during sequencing. The fidelity of the sequencer itself is only so high. So the top one is mutations in the DNA, apparent mutations observed in the DNA that are the result of the preparation for sequencing. This is what's called PCR amplification right there. And you get random errors which occur due to the sequencing process itself. We're only able to read DNA with so high fidelity with production DNA sequencing instrumentation. So because of that, you have a lot of noise. And one of the ways we correct for this, we'll stick with this page, is via what's called a unique molecular index. And what that means is we basically label each molecule we get with between 3 and 5 base string of DNA that we know about in the front of the molecule. So that way, what we can do is when we amplify this molecule for sequencing, so we have to copy it many, many times for sequencing. we're able to trace it back to its parent lineage. So we know the original molecule. And what that allows us to do, especially in the context of these PCR or sequencing-induced artifacts, is create what's called a consensus. So there are a number of different algorithms we use to create a DNA fragment consensus based on the unique molecular index present in all of those molecules. So we know all of those molecules came from the original parent molecule. We know they must all have the same sequence by virtue of the fact that they all came from the same molecule. And then the subsequent act is computing their consensus sequence and collapsing it down into a single molecule. There's where you see the error correction and quantification methodology. By the way, happy to go into more depth on any of these points if anyone is interested. Feel free to stop me. One of their technical challenges is it's difficult to accurately estimate circulating tumor DNA levels with a small number of alterations. Because we're sequencing very deeply, we can't sequence very broadly with breadth. So it's not always easy to estimate how much ctDNA is present in the patient. There are a variety of different methods you can use with looking at the mutations or alterations you detected. But as you can see, both of these methodologies are pretty terrible. They don't correlate well with what we would expect with what we know CTF circulating tumor fraction to be. This is based on high fidelity, comparing these various methodologies with a high fidelity methodology that is low pass whole genome sequencing is what it's called. So what we see is it's very difficult to get a good estimate of how much ctDNA is present. Those early plots showed very cleanly, but they had a number of advantages that a standard production sequencing methodology does not have. So it creates a lot of challenges there. So yeah, to that end, we created the Tempus XF assay, 105 gene targeted sequencing assay. Clinically detects short variants, copy number alterations, gene rearrangements, and microsatellite instability. And we have a variety of specialized bioinformatics computational pipelines for error suppression, circulating tumor estimation, and a number of other features. We also do some interesting stuff with combining this test with our tissue oncology offering, the XT assay, because then we can gain knowledge combining our tissue sequencing and our liquid biopsy assay about how to better treat a patient. So yeah, here going into a little more detail on how we're using unique molecular indices to suppress errors, discussing consensus sequencing, and then we have a little better information how we take. Can you all see my mouse? Can you see my mouse? Okay, perfect. Our unique molecular indices on both sides and are able to collapse them and suppress errors. We see these error bases here. The true molecules here, you could collapse them at a single-stranded level, and then you can collapse them at a double-stranded level because, of course, DNA is reverse complementary. So we would expect a forward and reverse fragment to agree on the presence of a genomic mutation. So this is one of our consensus methodologies of resolving. It's called duplex single-strand consensus or duplex consensus sequencing, which is one of our computational error suppression methods. We also have error rate as a function of nucleotide context. And that is to say, when you're doing DNA sequencing, what you're doing is you're actually having a DNA polymerase, like all prokaryotes and eukaryotes have inside them, how our own cells replicate, copying the DNA. And that polymerase has different fidelity based on the nucleic acid base it is copying and the surrounding nucleic acid bases. So here we're seeing the error rate in a context-specific manner. So what we see is certain genomic contexts actually increase the error rate a very significant degree. We're seeing a GCC context or a GCG context, that is to say when the polymerase is trying to copy these bases, has a much higher error rate than a CCT context. So our error rate is both non-random and variable. Error is non-random and it's variable based on context. So that's to say error can be position-specific and context-specific, which creates a lot of problems in detecting mutations, especially mutations that are in these difficult to these error-prone contexts. So, how to address this? Again, this is in the paper. If anyone wants to go into a little more detail, each error context can be modeled as a beta binomial distribution. all trinucleotide contexts that exist. So there's three nucleotides in a trinucleotide, and there's 4 possible bases for each trinucleotide. So there's 3 to the 4th trinucleotide context. So first, what we do is we compute an empirically derived error model from a variety of putatively healthy samples. So essentially, we get a bunch of blood draw, what have you. We do a blood draw of healthy volunteers. We sequence them, and we compute an empirical error model for each base. That is to say, the error rate at that genomic position. That again is modeled as a beta binomial. And then we have and then we take the trinucleotide context. We provide the same methodology to compute the error rate within a context-specific manner applied across each trinucleotide context present in our assay. So we have a hierarchical model which applies the beta binomial distribution based on the local error rate and sequence context. And in that way, we're allowed to suppress a lot of these errors based on our empirical observation and the application of the hierarchical application of these models. So yeah, as it says there, sequence large panel of healthy human normals, compute position and trinucleotide specific shape parameters. So of course, your beta binomial distribution has your alpha and beta shape parameters. In this case, we're using the method of moments. There's a number of different methods you could use, but using a variety of ways to optimize our shape parameters. And here's what we see. This is work from a group at Pfizer, if I recall correctly, where they're showing a similar methodology and its performance compared to a variety of different other error correction methods. So first they have raw, which, as you can see, there are no error-free positions. If you do not do error correction on high-def data, every base will have some errors. It will have some error rate in it. If you do barcoding, you can suppress these errors significantly. You can get up to 60%. So sorry, this is y-axis is error-free positions percent. X-axis is patients. So these are patients, and we're looking at the error rates within each of these patients. And then we see 2 different error suppression methods. One that takes the top one, TNER, takes into account trinucleotide specific context. The IDES method does not. And we see us moving closer and closer to 100% of positions being error-free. And using these empirical distribution models, these learned empirical distribution models, we are able to suppress errors to a very high degree in our high-def sequencing data. We then also, at Tempus, we have a vast database of solid tissue oncology specimens. So our big question here was, how do we take what we already know about mutations in cancer and apply that to detection of very low frequency variants? So to that end, we employed a Bayesian framework for on top of our error suppression framework for calibration of variant detection of variant detection thresholds based on cancer diagnosis. That's just a fancy way of saying if you're a patient with a cancer type that has a high prevalence of mutation X, we lower our thresholds for detecting mutation X. And the way to do that was essentially taking a very basic version of this model. That is to say, what is the post-test probability of a variant being present based on pretest information and what we've observed. So it's an extremely straightforward application of Bayes' theorem to the process of variant detection, and it's proved quite effective in allowing us to calibrate our performance based on what we already know. We've seen so many thousands of patients of different cancer types in our solid tissue assay. We know what their variant distributions look like. We can apply those variant distributions on a prospective basis and calibrate our software to better account for what we expect to be there. And calibrating our pre-test odds for a given genomic event allows us to modulate our specificity requirements based on our prior. And that's, it looks very straightforward when we write it here, but there was actually a lot of work that went into it. But essentially, this is another method we use. If you'll learn one thing about me, it's that I'm a Bayesian. I feel very strongly about Bayesian methodologies because I feel like it's very important for biology. Just because we have so many knowledge gaps in biology, it's hard to employ strict methods without saying, hey, what do I know? What do I don't know? How do I use my prior knowledge? But yeah, as you can see, two different methodologies that largely rely on Bayesian analytical frameworks. This one's a little more classical. So we also worked on, along with our suppression methodologies, we're also talking about our methodologies of estimating circulating tumor fraction. So as we showed earlier, it's actually quite difficult to estimate circulating tumor fraction just by looking at circulating mutant molecules. So what we did was we took what I would consider a bit of a sensor fusion-based methodology. We took large-scale genomic alterations that we can detect, so structural alterations or copy number alterations, and we integrated them with our variant data to create a combined data stream estimating, performing a maximum likelihood fit of circulating tumor fraction based on what we observed both on the structural side and on the variant side. So here is what an example of what I'm talking about. So here's a tool that's used by other groups. It's called iCourse-CNA. Was developed by the Broad Institute, if anyone's interested. And what we're looking at is a human genome. This is a human genome derived from liquid biopsy sample. You can see in the top, the tumor fraction is 0.2692. That is to say, 26% of circulating molecules are estimated to be tumor derived in this context. We see a number of interesting events. The blue indicates copy neutral for those who have not. Let me take a step back. If this is your first time looking at copy number plot, every human has two of each chromosome, except for sex chromosomes, of course. So the expectation is that everyone should have two copies. And this line measures the deviation from expected copy state, which is to say two. You should have one chromosome from your mom and one chromosome from your dad. So in that context, you see many of these are copy neutral. They're normal chromosomes present in the cancer. And then we have here where we've seen a loss of chromosome 7. So instead of having two copies of chromosome 7, there's one copy of chromosome 7. And for copy number analysis, what we can do is estimate the percentage of genomic material that has a cancer-derived copy state. So because this is essentially an integer best fit problem, you have a discrete integer count of chromosomes. If you have two in each of your cells has two copies of chromosome 7. Obviously, cell division is a different topic, but on average, you have two copies of chromosome 7. You can have two copies or one copy. You can't have 1.5 copies. So because we know this, we know you have an integer number of chromosomes. We can compute the mixture proportions from the deviation from expected integer counts. So in this context, what you'd see in a pure specimen is this one copy state would be very far down around here, actually around -1. This is a log2 ratio plot. But we know that the patient has the patient's regular cell-free DNA that is not cancer-derived has two of every chromosome because they're putatively a normal, healthy human. So we're able to say, okay, we have a mixture of copy states available. We have some subset of the sample has one copy of chromosome 7, and some subset of the sample has two copies of chromosome 7. What is the mixture proportion that fits our observation? And this is where you can employ maximum likelihood fit approach to compute the tumor mixture proportion based on the observed copy states. And that's what a lot of tools do at this scale. So you can say, okay, the best fit to our observation would be this mixture proportion, and we can use this information to get a very high resolution copy state information. That being said, our panel is not as broad as the low pass whole genome sequencing data. Our panel is 105 genes, so we have to use a variety of methods to accommodate the fact that we don't have the same breadth of resolution. And yeah, this is the low pass whole genome sequencing method. As I said, the disadvantage here is that it requires a whole separate laboratory protocol. So we wanted to figure out how to do this, but without using that separate laboratory protocol. So what we did is leverage a very interesting property of hybrid capture sequencing, and that is that hybrid capture sequencing is not 100% effective. It's about 70-80% effective, depending on efficiency of the reaction. And what that means is you have essentially a off-target reference genome. So anything that's not on target from the hybrid capture reaction comes from everywhere else in the genome. So we can essentially bootstrap a whole genome from that information, from the differential dosage of that information from off-target reads. As it says here, off-target reads are distributed randomly throughout the genome, effectively low-pass whole genome sequencing. So it was very fortunate that that's true. So we developed a tool called Otter, which uses off-target coverage data to estimate copy number, dosage, and circulating tumor fraction. And here we see our plot. And if you look at the earlier plot, which was looking at mutant molecule-based approaches, you can see extremely high correlation between our method and low-pass whole genome sequencing, to the degree where we felt it would be an adequate replacement for the technology. So how do we use these features? So we've discussed a number of different technical methodological improvements we've developed to our XF panel, or to solve a number of problems of liquid biopsy in our XF panel. And now we want to use these to help patients. One big one for us is concurrent testing. That is to say, we test patients with XT and XF, our solid tissue assay and our liquid biopsy assay, either at the same time or at subsequent time points to monitor disease progression or therapeutic response. This gives us a number of different advantages, of course, with tracking. We also are able to disambiguate hereditary alterations, and it allows us to detect, this is a bit of a doozy, what's called an event called clonal hematopoiesis of indeterminate potential, which is essentially somatic mutations that aren't from the cancer, but from a pre-blood cancer-like state, a pre-leukemic or myeloid dysplasia or myeloid state, where essentially a lot of patients, especially as they age, get very low frequency somatic mutations in their white blood cells. And they often look exactly like mutations that we'd see in cancer, so they can be a significant confounder to analysis. So using this tumor tissue and liquid-based methodology, we can disambiguate these variants that we would normally think would be from the cancer, but are actually from a wholly different process. As you can imagine, we don't want to make therapeutic recommendations based on variants that are non-cancer derived. So you can see in this graph, there's a number of different variants that are yellow dots, where we're actually able to distinguish them as non-cancer derived. So since we have the solid tumor, we know they didn't come from the solid tumor, and we can say, hey, this variant came from somewhere. We don't know what to do with it, or we should not include it in our analysis. And then here we see liquid-solid concordance, a variety of pluses and minuses there. Also on the same side, we're looking at mutational profiling. Here we see distribution in our 1,000-patient cohort of mutation types. Of course, we recapitulate what you'd expect to see in a population of large solid tissue tumors. And then we see circulating tumor fraction levels by cancer cohort. Not a lot there, it's mostly interesting plot, but we see slight elevations between the different, slight elevations and depressions between the different cohorts, but we're allowed to very, very accurately profile that. And then of course, with this accurate CTF estimation, we're able to longitudinally review patients and look at how their cancer is, look at how their circulating tumor DNA levels go up and down over time. And here we see a number of different patients, all starting from 0 in this graph, and then we see days after first sequencing. And this is the relative deflection of that patient with their original biopsy time, liquid biopsy time point. So we can see patients going up, patients going down, patients with fairly zigzaggy plots. And all of these indicate different therapeutic response dynamics, which can be useful for the physician to track. And of course, yeah, this is the information that really drives a lot of treatment decision making, is what are the ctDNA dynamics looking like? And then we can also use these methods to monitor for progression. What we clearly see, I mean, at a baseline, how much ctDNA you have is a strong indicator of whether your disease is progressing or not. Here we're seeing correlation of circulating tumor fraction level by RECIST criterion being a radiographic scoring method that oncologists and radiologists use to indicate whether a disease is progressing or not. So you have progressive disease, partial response, stable disease, complete response. As we see, the vast majority of patients we sequence are in the progressive disease phase, and progressive disease very strongly correlates with high levels of circulating tumor fraction. So this is a good indication that we can say, hey, this patient needs to be seen by their oncologist. We need to make therapy changes, make decisions about what needs to be done here. This high resolution CTF estimation can drive those sort of conversations. Conclusion. Yeah, despite the technical challenges, liquid biopsy is a valuable tool for physicians to monitor therapy administration. I'm very proud of the platform we've developed at Tempus. I think we've overcome a lot of very interesting technical challenges with fun computational methods that we can use to improve patient care. And yes, these methods can be integrated very effectively with other diagnostic modalities like tissue testing or radiography to maximally inform oncologists on how to treat their patients, especially in the advanced disease setting. Finally, I want to thank, of course, my team, the Bioinformatics team at Tempus, the clinical science team. The clinical science team is both the medical team and the our variant and disease curation team, and then of course the molecular R&D team that's our counterparts on the chemistry side. None of this would be possible without them. They've done amazing work. I'm very, very proud to work with them, and it's been a wild ride. So yeah, thank you all for your time, and I'm happy to take any questions you might have.